Okay, after looking at the dengue virus, we'll be looking at the dengue disease, right? The disease is a tropical disease because the dengue virus is transmitted by Aedes mosquito, which lives in tropical areas. Okay, so I'm sure you have some familiarity with the symptoms of dengue fever. Okay, so if dengue fever is not treated early, right, it can develop into more serious cases known as dengue hemorrhagic fever as well as a dengue shock syndrome, right, which is due to the low level of platelets. So there's like the inability to stop bleeding, especially internal bleeding, right, and that brings about the low blood level we see in the dengue shock syndrome. Okay, so the Aedes mosquitoes are the ones that transmit the dengue virus. There are two species, right? One is the Aedes aegypti, which we are more familiar with and more concerned with because it is the one that is more adapted to live with humans in the urban environment, right, like Singapore. Okay, but for the rural environments, right, there's another species known as Aedes albopictus, which transmit the virus. Okay, so we'll then look at the life cycle of Aedes aegypti, right? So it has a 4-stage life cycle, like many insects. The first is the eggs, right? So the female mosquito are the one that will suck our blood, okay, in order to gain enough proteins and energy to make eggs and lay eggs. Okay, so they would lay eggs in usually in an urban environment, where they will lay eggs in the inner walls of water containers. And the eggs are very hardy. They can stay dormant for up to 9 months if the condition is not favorable. Which means, right, even if we pour away the water in the containers, right, if we don't clean the containers thoroughly by scrubbing the inner walls, right, the eggs can get stuck, can remain stuck on the inner walls of the containers. So the moment there are water, right, collected in the containers, then the eggs can hatch as long as the eggs are still alive, which can be for a long time, right? Then once the eggs hatch, right, there will be the larvae, which stays at the surface of the water because they still breathe air, right? They don't breathe dissolved air in the water. Right? There's a part of like a tube-like structure in the body that breathes in air above the water. Okay, so you do see them if you look at the water, right? You can actually see the larvae moving around. Okay, and the water needs to not be that clean for the larvae to find food, right? Because it feeds on organic matters, certain microscopic organisms, right? Algae, or even dead organic matters, right? And then, right, the larvae would develop into pupae or pupa, right? So these are not mobile anymore. They still stay in the surface, at the surface of the water, but they are not mobile. They don't feed. And after about 2 days, the mosquitoes would emerge from the pupa and, of course, leave the water. All right. So for the adult mosquitoes, on average, they live for about 2 weeks and can lay eggs for up to 3 times. Okay, but with global warming, this is changing actually, right? They are able to lay even more eggs, or lay eggs for even more number of times because of the rising temperature, which we'll look at a bit later. Okay. Right, then transmission of the virus. We have looked at this earlier when we looked at the dengue virus, right? So the Aedes mosquito is the medium, right, to transmit the dengue virus from an infected person to an uninfected person, right? So when the Aedes mosquito bites the infected person and suck up the blood, right, the virus would be transmitted to the mosquito. Then this infected mosquito would then transmit the virus to an uninfected person when it bites the next person. Okay, so when the virus feeds on an infected person, the virus, or when the mosquito feeds on an infected person, the dengue virus gets into the meat gut of the Aedes mosquito, because it sucks up the blood, right? And the blood contains the dengue virus. Okay, so it's found in the gut, or meat gut to be more specific, and the virus will replicate in the meat gut for a few days. This few days is called the incubation period. Okay, and the virus would then travel to the salivary glands of the Aedes mosquito. So the saliva of the Aedes mosquito would then be containing the dengue virus. Okay, so when this infected mosquito bites another person, it will inject its saliva into the person. And this saliva obviously contains the dengue virus, right? So the dengue virus gets into the next person that the infected mosquito bites, all right? And of course, then the dengue virus would start to infect the whole cells, right, found in this person, okay? So the virus would replicate inside this person such that, right, there will be high level of virus in the blood in this person. So if another Aedes mosquito bites this person, right, within about 4 days, this mosquito would be able to not just draw up the blood, but also the dengue virus as well, and this mosquito would become infected, right? And you see that the symptoms of dengue fever as a disease, right, only developed about after 5 days, right? So which is after the number of days where this person can transmit the virus, right, to another mosquito, right? So this infected person may not even know that he or she is sick, but then he or she is able to transmit the virus. That's what makes the virus and the disease so successful. Okay, so you have this diagram in the notes, right? If you take a look at the diagram, the number of days mentioned here, right, is a bit different from the number of days we went through just now, and also those in the text earlier in the notes, right? That's because the diagram and the information in the text came from different sources, from different countries. So with different countries, different temperatures, different weather, right, the number of days is a bit different, because these incubation periods and the time for the mosquitoes to develop also, and the virus to also develop, right, is affected by temperature. Okay, so yeah, so you don't have to be too concerned with the number of days. Just kind of know the whole sequence of events, right, will do. Okay, let's move on to how the viral infection, right, brings about the disease, right, of dengue fever. All right, so the dengue virus is introduced into the person's bloodstream, right, from the saliva of the infected mosquito. The virus infects dendritic cells and macrophages, as we have looked at earlier when we looked at dengue virus. And these infection, right, would be enhanced and amplified when the infected cells enter the lymphatic system. Okay, so what is the lymphatic system, right? The lymphatic system is like another set of circulatory system. I think I can say that, right? So the usual circulatory system that you have learned before in secondary school, right, involves circulation of the blood, right? And then you have all the arteries and arterioles and veins and so on, right? But there is another system of vessels that transport what we call lymph, which is something like plasma. So there's no red blood cells in it, right? So this is the lymphatic system. Its function is more for immunity rather than transporting of oxygen and food and waste and all that. Right? So it's a lymphatic system. It does not have the red blood cells, but it does have quite a high concentration of white blood cells or lymphocytes, right, like the T cells and the B cells. Okay, so the dendritic cells and the macrophages, right, that the dengue virus would infect, they can also enter the lymphatic system to interact with the T cells and B cells. Okay, so when the infected cells get into the lymphatic system, what happens is the virus is spread, right, to other parts of the body via the lymphatic system. That's number one. Number two is, again, we have some dendritic cells and macrophages in the lymphatic system too, right? So when the infected cells get in, then the virus that is going to be released from the infected cells would be easy, make it easier for it to find another dendritic cells and macrophages to infect. Okay, so it's easier to find the whole cells in the same lymphatic system, and also the virus gets spread via the transport in this lymphatic system. Okay, that's why we say that the infection is enhanced or amplified, right, when the infected cells get into the lymphatic system. All right, so we are back here. Okay, so when the infected cells and the virus, right, is transported to different parts of the body, right, it comes into contact with different organs. So it has been found that besides the dendritic cells and the macrophages, the dengue virus is in some cases also able to infect cells in the organs, right, such as the spleen, liver, and bone marrow, right? So it's quite broad in terms of the type of whole cells the dengue virus can infect. Okay, and in the serious cases, right, the dengue virus will be able to infect the megakaryocytes that produce platelets. So when the megakaryocytes dies due to the infection, then there would obviously be less platelets and less blood clotting, which requires the platelets. Right, so therefore that leads to the dengue hemorrhagic fever and dengue shock syndrome, which are the serious cases of dengue fever that we have looked at the start of the video. Right, so that was about infection and disease, but obviously our body has protection, right, immune response against this infection. Okay, so as we have looked at earlier in a previous video, right, infected cells would be able to release cytokines, right, to tell the immune system that they are infected, right? So the immune system would be able to then kill the infected cells. Okay, so the type of immune cells that specifically kill infected cells is known as cytotoxic T cells. Right, there's another kind called natural killer cells as well. So these are cells, immune cells that kill infected cells when the infected cells release cytokines, right, to tell the immune cells that they are infected. So obviously when the infected whole cells die, then that stops the replication of the viruses, right? And also our immune system is able to learn and remember previous infection, right? So if we have been infected with dengue before, then our immune system would produce antibodies specific to this dengue virus. So if we are infected by, say, serotype E1, for example, right? The next time we are infected by E1 again, we have antibodies against this particular serotype of dengue virus, which would be able to bind to the virus, particularly on the glycoprotein E, right, to block infection, right, block attachment and block the entry of the virus. Okay, so we do have some defense against infection. Okay, but right, if you are thinking clearly, the dengue virus infects immune cells, right? Specifically the dendritic cells and macrophages. The function of the antibodies, of course, number one is to block infection. The functions of the antibodies also, right, increases the efficiency of immune cells phagocytosing or ingesting the virus. Okay, so when the antibodies bind to the virus, right, it blocks infection, obviously. The antibodies are also like tags, right? Tags to tell the immune cells, especially the dendritic cells and macrophages, that this is a virus, come and eat. Right? So that increases the efficiency of phagocytosis of the virus by the immune cells. Okay, so the dengue virus infects immune cells. Antibodies makes it easier for the dengue virus to enter the immune cells, right, via phagocytosis. Do you see a problem here? Right? So if the antibodies are not functioning properly, right, maybe it doesn't really block the viral infection properly, or it doesn't prevent the entry, right, the fusion between the viral envelope and the phagosomal membrane. If it cannot block that properly, yet it can increase the efficiency of phagocytosis, which is how the virus enters the immune cells, right? That actually increases the chance of infection, don't you think? So keep this in mind, right? We'll come back to this problem a bit later. Right, next up, we'll talk about the susceptibility of humans to the virus and therefore the virulence of the virus. Okay, so the virulence, how easy it is, right, for the virus to cause disease, how severe the disease is going to be, right, can be affected by a few factors. Right, number one is the serotype. This one we don't really know why, but at least in this case of the epidemic in Cuba in 1981, it was found that the serotype 2 actually gave rise to a more severe disease, and there's more people dying. We don't really know why. Okay, maybe because the immune response against this serotype is a bit less. Not sure, right? But the principle basically is that different serotypes may give rise to different virulence and susceptibility, right? Secondly, right, it's called the second antibody-dependent enhancement, right, mentioned here, which comes about if we have a secondary infection by the dengue virus of a different serotype. Okay, so remember the problem with antibodies just now, and remember that there are different serotypes of the dengue virus in which the glycoprotein E is slightly different. Right, so if you have been infected, say, by serotype E1 before, right, then you have antibodies against this serotype. So if you are infected by the same serotype again, the antibodies should be able to bind to the glycoprotein E, block entry of viruses, block attachment, block infection, right? No problem. But what if you are infected by a different serotype, say serotype E2, for example? The glycoprotein E is similar to E1, but not exactly the same, right? Slightly different. So there may be chances that the antibodies that you have against serotype E1 is still able to bind to the virus of serotype E2. So it can still bind, it can still tag the virus and tell the immune cells that this is a virus, come and eat. But yet, because of the slight difference in the glycoprotein E, the antibodies may not be able to block completely the attachment and entry facilitated by glycoprotein E. Right? So it makes the phagocytosis, which is the entry step, right, of the virus into the immune cells, easier, yet it cannot block completely the entry, right? So maybe the immune cells, say macrophages, is able to phagocytose the dengue virus tagged by this antibody much more easily, and then the dengue virus is placed in the phagosome. So by right, the antibody should block the fusion between the viral envelope with the phagosomal membrane, but because of the difference in the glycoprotein E, maybe this is not blocked. So the virus is actually able to continue with the entry step and therefore continue with the rest of the reproductive cycle. All right, so this is known as antibody-dependent enhancement, and we observe this, right, when we observe individuals who are infected a second time by a different serotype of dengue virus. All right. So thirdly, right, the time period between the first primary infection and the next secondary infection also play a part. Right, the longer the interval between the two infections, the higher the risk of a more severe disease, probably because the longer it is after the first infection, right, even though our immune system should remember the encounter and there should be antibodies, right, the number of antibodies might drop, right, and the immunity against the dengue virus due to the first infection might wane after a longer time, right? So therefore, when we are infected by dengue for a second time, it may cause a more severe disease. Okay, so these are some factors that affects the susceptibility and virulence of the dengue virus. Okay, so last part, we will look at a possible drug target, which currently scientists are looking at. Okay, so this quite obviously, right, would be the glycoproteins that are involved in attachment and entry of the virus, right? And anyway, these are the structure of the virus that are found on the outermost layer, right? So drugs or antibodies that bind to the virus should bind to them, right? So these are the most obvious drug targets, right? But the problem is, especially with glycoprotein E, is that there are the 4 different cell types, right? So drugs that are able to bind to one may not bind to another, unless you can find the similar structures between the 4 different types of glycoprotein E's and make one drug that's able to bind to all 4 different types. So currently that is still under research as far as I know. A second problem with developing drugs is, of course, the high mutation rate of the virus. So remember that dengue virus, part of the reproductive cycles requires RNA-dependent RNA polymerase. And we have looked at earlier, right, that RNA-dependent RNA polymerase is more error-prone and doesn't have the ability to check for errors, right? So that's why there's a higher mutation rate. So mutation will cause even more changes, right, to especially the glycoproteins, right? And that renders current drugs, if any, may not be so efficient, right, in the future. Okay, right.